We're now going to go on to the next element of our program. One of the things that we promised you at the very beginning of our broadcast, or maybe I should call it a Zoomcast today, is that we are bringing you world-leading experts, and that means from around the world, including now returning to my former country, and I'm still a citizen as well. I'm a dual citizen, an Aussie and a Yank, as they say here. But we're going to go deep south to Matt Might. And Matt Might is the director of the Hugh Call Precision Medicine Institute at the University of Alabama, Birmingham. He is also on the faculty of Harvard Medical School in the Department of Biomedical Informatics. He was a strategist in the executive office of the President of the United States in the White House. Now, just in case you're wondering about his politics, don't, because that was in both the Obama administration and the Trump administration from 2016 to 2018. So it is fair to note and to pay attention to the fact that rare disease is distinctly apolitical. It happens to all of us, no matter who we are, where we are. Matt's breakthroughs have been written up in the New Yorker and the New York Times. He is also a father, a member of our community. He knows deeply the perils, the real hardships, indeed sometimes the tragedies, as well as the triumphs of the odyssey that you are embarked on with your families. And I would encourage you, if you have time, for everyone on this Zoom call to make sure that you look up Matt Might and watch the video on why he became involved in rare disease. And that will tell you more about his son, Bertrand, and NGLY1, and also about his personal experience with this. But today, what we're going to hear from Matt is about a broader picture as well. I remember meeting you, Matt. I don't know if you remember this, but I remember meeting you back in 2014 at a KCNQ2 Cure conference. And I think you were just at the White House then, starting there, and you had done a couple of really stunning posts that had brought attention to rare disease. And you had also written a story for the New Yorker. What do you do if your child has a condition that is new to science? So much has happened in the years since. At that conference, you told us that you and your house were the FDA deciding on some of the medicines for your child. You went on to create some of those. And I want to thank you for joining us as a parent, as an absolute world leader in precision medicine, to tell us about the algorithm for precision medicine. Ladies and gentlemen, and all the kids on this broadcast too, welcome to Matt Might.
Well, thank you so much for having me. And yes, I do remember meeting you in DC. I think it was 2016.
That's right. Oh, you're right. It was 16.
It was, yeah. So if it's okay, I'll just share some slides, and I will actually share my personal story at the end as an example of the algorithm for precision medicine. But my gosh, it is such a pleasure to speak to you all today. So you assume you can see my slides, right? Okay. All right. Well, I have to start with a disclaimer before I present the algorithm, which is to say that if there is anything in this talk that resembles medical advice, please, for God's sake, do not take it. I don't have any medical training whatsoever. I am a computer scientist. I still spent most of my career as an academic computer scientist, and got pushed into precision medicine professionally because of my son. And I'll tell you about his story towards the end. I've still to this day spent most of my career in things like cybersecurity, high performance and supercomputing. I did do a little bit of medical device verification back in the day, but mostly it's been in fundamental research into algorithms and data structures and things like that. So today I want to get across three things. I want to tell you, first of all, what precision medicine is, because this is a field that holds a lot of promise for all of rare disease, and especially for rare genetic epilepsies. I want to explain what I call the algorithm for precision medicine, this step-by-step process that we now apply in my institutes for taking patients on their therapeutic odysseys. And then I want to show you some examples of what it actually looks like when you do it in practice. And I'll close with my personal story of how I got into precision medicine. And yes, my son did have a rare genetic epilepsy, and that became a large part of my life's mission for a very long time. So what is precision medicine? Well, if you trace it back to its origins, it's now, I think, formally about 10 years old. The National Academies of Science released this report about 10 years ago. And there's an essay in the back in the glossary defining this term, precision medicine, trying to say what it is. And if you really boil it down to what they really meant, what they're saying is that if you are using the specific characteristics of a patient to tailor medical treatment to them, then you're doing precision medicine. So it's also kind of like personalized medicine, except that there's an emphasis on getting really molecular resolution when it comes to the individual characteristics of the patient. And then a few years later, President Obama launched the Precision Medicine Initiative. And once he launched this, there was this explosion of competing definitions for what precision medicine really was. And I honestly spent a lot of my time and since then cleaning this up, trying to come up with a common unified definition for the whole field. And what I've distilled it down to at the end is that precision medicine is really all about optimizing health with data. So if you're using data to optimize health, you are in some sense doing precision medicine. And I think one thing that makes this a little bit different than previous attempts at optimizing health is the amount of data that's often available on patients. And so, for example, if you're going on this process of trying to find the best possible treatment for a patient, you have tremendous data at your disposal today that you just didn't have 10, 15 years ago. For starters, you have clinical access to the entire human genome. That wasn't possible not long ago, but this is an enormous piece of data. And sifting through it to find the mutations that could be driving a single patient's epilepsy absolutely requires computation. It's just too big for a single physician to sort through all that data. And then, of course, there's already the longitudinal electronic medical record data that's available on patients. And honestly, I would say we're still not fully exploiting this when it comes to helping patients. And then I also include all the data outside the patient. So all the data locked away in scholarly articles and textbooks and biomedical databases, all of which could be part of this optimization problem for the patients. And by optimization problem, and this sort of belies my engineering background, I really do think of it like an optimization problem where you use all the data to form these constraints and look for the combination of lifestyle and treatment that achieves optimal health for that particular patient. So that in a nutshell is to me what precision medicine really is. Now, another kind of disclaimer here. In practice, we often don't have enough data to jump all the way over to the best possible treatment for a patient. Oftentimes, we will have to do some scientific research or an experiment on their behalf to get more data to get us closer to the best treatment for them. And in practice, and in sort of full fairness, I would say in my experience at the Institute at UAB, that maybe 10% of the time, we can make a pretty good guess as to what might help the patient right out of the gate with nothing more than the data that they bring to us. 90% of the time, we're going to end up going to the bench to do something to try to get closer to that prediction for them. And this is really what I spend most of my days doing. I was asking NIH to fund the infrastructure to do this kind of research for every single patient that reaches out to us. So I do think of it as a process, really a two-part process. There's a precision diagnosis portion of precision medicine. There's a precision therapeutics portion of precision medicine. I'm not going to talk a lot about the precision diagnostic portion, even though that's very important. It was very important to me personally at one point in time. I am most excited about the precision therapeutics side, and that's where I spend a lot of my energy. How do you go from that molecular diagnosis over to a treatment? How do you find the drug that stops the seizures in this case? And so what is precision therapeutics? And in the end, at a very high level, I think there's two main approaches, and I'll break both of them down for you. They're the rational approaches and then the empirical approaches. And in the rational, you're trying to take a very first principles approach, a root cause approach, saying, here's the molecular cause, and this will be the drug that inverts this molecular cause, this molecular mechanism of harm, or something like that. And then there's the empirical, where you just build a model and try a lot of stuff and see what works at the end of the day. And both are actually applicable to rare genetic epilepsies. So in the empirical approaches, what really happens here is you're starting off with your causal genetics, and you're trying to find the best possible treatment. In the middle of this is bench work. You're going to be doing experiments. You might work with cells or animal models or test drugs. And from these experiments, you want to find that medication. So in a little more detail, here's what this process looks like. Start with the patient and you extract a model. In this model, it could be the patient's actual cells. That's certainly a possibility. But it could also just as well be an animal model, or it could be a cell model that's constructed from a different cell line, but where the patient's mutation's been added in. There's lots of ways you can build a model. All that really has to happen here is you have to be able to observe the disease in that model. And what we call a phenotype, just think of it as something that tells you that this model isn't well, and it is accurately representing the patient's disease. Once you have that signature, you can test drugs on the model and check to see if anything corrects the signature and gives you, a rescue in the model, or essentially a healthy patient, or a healthy model. And hopefully, if you try a lot of drugs against the model, at least one of them will rescue. And then the hope is that one will also work in the patient. Now, it's not always the case that a rescue in the model means a rescue in the patient. And I'll tell you the biggest stumbling block right up front. So there's no mystery if you go through this process. You will frequently find hits in the model that won't work in the patient for the following reason. The concentrations that work in the model could be, in some cases, orders of magnitude off of what you can actually safely dose a human being. And so, just because you see a rescue in the model doesn't mean you're done. It means you have to do this whole safety check, make sure that those concentrations are achievable, make sure that the drug will actually get to the right parts of the body. So you might need a drug that gets in the brain. So you've got a sort of a concentration issue or an efficacy issue. You might have a targeting issue, and then if you solve those, you're good to go. Now, so in some cases, even if you test all the approved drugs in the world, you might have a little bit of medicinal chemistry left to do to get there. And this is actually a great approach for epilepsy. And that's because, not all epilepsies, obviously, but a large number are driven by ion channels. And these tend to be open to pharmacological modulation. On top of that, they're measurable. So, there are ways that you can actually measure the effect of drugs on ion channels. So that you can actually study them in a dish, on cells, in fact. And so this gives you a way to actually screen drugs. On top of that, if you move to something like an animal system, animals often have seizures. So if you Google around, you'll find these great videos of animals like zebrafish actually having what appear to be their version of seizures. And so you can use this from all the way from cells to animals. You've got ways to check for the behaviors and the phenotypes of interest in these models. So another term for this field is coming to be precision disease modeling. And in fact, we've just established a new center inside of my institute for doing exactly this. We call this the Center for Precision Animal Modeling. What we do is we accept from physicians genetics reports. We use that to create a model system, some kind of organism that has a precise recapitulation of a patient's disease. It creates essentially a personal precision avatar for that patient. It's a pretty beefy award in terms of the infrastructure they've given us. It doesn't include enough to do all the drug screening on top of it, but it does give us the ability to at least create these models for patients as they reach out. And that is the platform that they need to go on and do drug screening after that. But we can look for modifier genes with genetic screens. We can test a lot of drugs against these models if we've got a supplementary funding source to do that for the model. And so I'm trying to build the infrastructure to do the things we need to do on a regular basis in precision medicine. And of course, there's companies now that are trying to get into this and provide this kind of service directly to patients as well, which is quite exciting. And then there are the rational approaches, and the reason I like the rational approaches is that as a former card-carrying computer scientist, these are very amenable to computational approaches. So you don't have to touch a bench. And this in practice makes them much less expensive, although they're not quite as powerful as some of the empirical approaches. Now, at the heart of the rational approaches, what you're trying to do once you have that core diagnosis is figure out what the true mechanism of harm in this disease is. So you're trying to figure out, in this case, what the mutation or mutations are doing to a specific gene. Are they making it overactive? Are they making it underactive? Have they knocked it out? Or has it become toxic in some way? So if it's overactive, you want to inhibit it. If it's underactive, you want to activate it. If it's gone, you need to compensate for it, or you need to replace it in some way. And if it's toxic, you want to eliminate it. So there's different strategies behind each of these boxes, depending on what you figure out is going on with that mechanism. And this really does lead to a role for computation or artificial intelligence. The tool that we've been building in-house, which is freely available, is called Metagenome. It's a tool that's built as part of this consortium project at NIH called Translator. So we're an AI tool that absorbs hundreds of data sets that are being accumulated from biomedical resources by this consortium, and we're reasoning over these to look for very targeted treatments on a per patient basis. So what is this program Metagenome? Well, in a nutshell, it's old school logic fused with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight. That's really what we're after with Metagenome. So you can ask it very low-level questions. You can say, with all the knowledge sources you have access to, can you figure out if there's an inhibitor for X? So that's a very low-level question. But you can also ask it very high-level questions, like what might be a treatment for this disease right here? And for example, if you ask it specifically about overactive bladder, it will make 145 different suggestions along with explanations for those suggestions as well, I might add. So how do we do this? Well, we've had to read 30 million papers, but that's what grad students are for. And now, of course, we didn't actually read all 30 million ourselves. We used artificial intelligence. We used natural language processing and tools that other people have built to do this kind of thing and absorb all these abstracts into a virtual brain. And then this translator community has added in many more data sets on top of that. So if you poke inside the virtual brain, here's what it looks like. It's got a bunch of nodes in it, these circles, and these circles are nouns, and they represent things like genes or drugs or diseases, or, concepts within biomedical research. And the edges between them are verbs. They're predicates, and they connect these nouns together. So, for example, you might find these nodes, Prevacid, which is a drug, and Engase, which is a target, and a relationship between them. Prevacid inhibits Engase. And so each edge essentially encodes a sentence, a piece of a unit of knowledge. And from this, you can actually start to make inferences. So if you have node blue increasing node red, and node red decreases node green, you can infer by increasing a decreaser that you're ultimately going to decrease your target. So you can hypothesize that you can have these indirect effects. And so you can unlock hidden knowledge that's out there that's not explicitly stated. So the brain for Metagenome is this giant soup of facts, all strung together with these edges and these relationships. So how do we take this to patients? Well, we follow a step-by-step process. We built what we call the algorithm for precision medicine. Towards the end, I'll give you a link so you can go browse a 90-page guide that I've constructed for patients if they want to follow the algorithm for precision medicine. And it's what we follow at the Precision Medicine Institute. But the goal of this algorithm is to constantly find the next step in the process on the road to identifying a therapeutic. And I'll show it to you in very high speed. So there's a diagnostic portion, there's a therapeutic portion, and really in the middle of this is that split I showed you earlier, where you've got a mechanism in the middle, and you're trying to figure out, is it overactive, is it underactive, should I build a model organism? All of that's in there. And the goal is to find the right medications. We've tested this out now on hundreds of patients that have reached out saying, please help. And now really we're shifting from proof of concept over to proof of scalability. Can we scale this up? And it turns out there's a secret ingredient in the scaling, and it turns out to be people, in particular, really good students. So we use undergraduate students to serve in the middle of this process and guide patients through it. So they're very heavily trained and supervised. But what they do is they route between the physicians, the scientists, the computational tools, and the patient, trying to continuously identify and push everyone towards the next step in the process. And I'll just give you some examples of what it looks like. So this first one's actually in epilepsy. There was a patient that reached out with seizures. Turns out they had a variant in a gene called TMLHE, which in turn was deemed to be a loss of function in the gene, or I guess in this particular allele, which led in turn to a carnitine deficiency and hence a recommendation of supplemental carnitine. And when we checked back in with the patient after about 30 days, it appeared to have significantly reduced their seizures for this particular genetic epilepsy. This isn't an epilepsy, but it's still another striking example of this algorithm. In this case, the gene in question was MAPK8IP3. It wasn't clear exactly what it was at first, but in conjunction with a consortium of partners, ultimately we deemed it to be a haploinsufficiency in this gene, or sort of a partial loss of function. And then Metagenome said, oh, but I know how to boost the function in this gene. You can use retinoids, or better known as these vitamin A derivatives. And so we passed this recommendation back to the treating physician along with the research to support it. They recommended it to the patient. Patient went on. And about 6 months into this, in a disorder where this 5-year-old boy had never even stood up off the ground, suddenly he's actually not only just standing, though with some effort, he's starting to take some steps as well, which you can see here. So the mom was kind enough to allow us to share this video. It's quite striking, again, considering 6 months prior, he couldn't stand at all. And, when you're making, impacts on a more developmental time scale, 6 months is actually not a long time to wait in our experience. And then a year later, the progress becomes really dramatic. So even just between these two time points, when he's about 6.5, you can see his gait is far steadier after about a year and a half on this treatment. And in fact, you'll see him just in a second, dip down, push himself back up, then take some more steps. And it's night and day, compared to where he was at the start of this process. And we've actually been doing a lot of research on this particular gene, trying to really unlock what exactly is going on here with these compounds, and also looking for even more effective ones now that we have a hint of where to go. So he'll just pick himself right up. Would have been unthinkable for this child just 18 months prior. Next patient is not an epilepsy, but in the orbit thereof. This is from a hyperactive potassium channel. And after it was confirmed to be gain of function, of course, we say, okay, how do we inhibit this gene? Because it's driving 150 cataplexic episodes per day in this poor girl. And when we started working on this a few years ago, we did drop it into Metagenome. We said, hey, show me inhibitors for this gene. And it says, oh, I've got one for you. Methamphetamine, which I'm pretty sure is also very illegal in Australia, or I would venture to guess it is. So great suggestion, solid science. It actually does work to inhibit the channel, just can't actually give it to an 8-year-old. It also turned up some venoms and toxins, and we got really captivated by these. So, oh man, maybe we can turn a toxin or a venom into a treatment for an epilepsy. Wouldn't that be cool? In parallel with that, we did that, comprehensive repurposing screen I talked about earlier. So we threw the kitchen sink at this one. And in the process of this, 3 more patients popped up. And I spoke to one of the dads, and he goes, oh, by the way, we already have a successful treatment for this. I go, well, what is it? And he goes, oh, it turns out it's amphetamines. So methamphetamine wasn't wrong. We didn't need the methyl group on it. We just needed the amphetamine part of the methamphetamine. And it turns out in this case, lisdexamfetamine, this extended release medication used for ADHD, was what worked. Learning from this lesson, realizing we actually had the answer under our nose the entire time, we've added a structural similarity search to the tool so that, if we get a hit that's outside what you're allowed to give to a patient, you can say, find me the closest matching structure that is something you can give to the patient, and then we can investigate from there. When I last checked in with the patient community, I think there's up to 5 patients around the world now, and they've all been successfully treated with this drug. And we're talking about a complete cessation of these cataplexic episodes. It is a dramatic impact on these patients. In fact, I think the oldest is now getting ready to go off to college and wants to go to med school after that. Another milestone we've hit recently, which has me quite excited, is a suggestion that came from the tool, making it all the way to an FDA-approved trial. So this mom here, some of you may know, she's a wonderful mom, very aggressive, has a child with ADNP autism, and reached out saying, hey, can Metagenome do something for my kid? And at the time, it didn't have enough data sources to make a prediction, but we don't give up on anybody. And a year later, it did make a prediction. So I reached back out and said, hey, looks like low-dose ketamine might upregulate reactivate this ADNP. So I gave it to the chief science officer for her foundation, who did the vetting. And after a year of work with this foundation, with this mom, with these families, really digging into the basic science of this, doing some of the preclinical work, they came to believe that this was a real potential hit. So February 2020, they actually got the trial approved and prepared to launch at Mount Sinai when COVID hit. So they got substantially delayed, but by September they managed to get this trial launched. So awaiting the results, but we're hopeful that this will make a real difference for these patients. Last patient I'll talk about for this section is just an example of what you can do when it's not necessarily a genetic disorder, but you have access to these kinds of tools. So in this case, it was intractable cyclic vomiting. This poor young woman was at 19 having multiple episodes of vomiting per day. She was effectively trapped in the hospital. She was 78 pounds. And her father relayed that, look, we've tried everything. We don't know what's left at this point. We don't know what to do. We don't know how to get her out of the hospital even. So it's rarely the case that you've tried everything for something like nausea, because there's a lot of papers out there. 64,000 of them are on nausea, and 4,000 are on treating nausea. And 347 there's 347 distinct treatments proposed throughout these papers. So if you rank them by the number of papers in support, you get zofran up at the top, most popular anti-nausea drug. But if you keep going down the rankings until you get to the unknown known region of this graph, where these are things that have been published, they're just not widely recognized, you get some interesting stuff out there. So with 3 publications in support, you get nasally inhaling the vapors of isopropyl alcohol during bouts of nausea. So we passed the evidence for this back across the medical team. They said, well, she's definitely not tried that. And it turns out it actually worked. So the next 8 months, she didn't vomit a single time. And she kept going after that without vomiting as well. But something remarkable happened in 8 months. She was up to 125 pounds. More than that, she actually got married. She was out of the hospital, married her high school sweetheart, and moved across the country to be with him in San Diego. And we're grateful to her for being so willing to share her story about her engagement with the Institute. So I'll close by saying, why is it me you're hearing from right now? And you heard a little bit about this from Sarah at the start. And it all goes back to my son, Bertrand. I was a happy professor in computer science. I was just starting out actually when he was a year old, and ended up on a diagnostic odyssey, trapped on undiagnosed island, not sure why he was having, so many seizures per day. I think it was hundreds of seizures per day back then. Why he had extreme developmental delay, why he had, a movement disorder, and why, for some reason, he could not make tears. He just couldn't make tears. He could cry, of course, but he just never made tears with it. And this, of course, led to all sorts of eye problems. He had eye surgeries to, drain the pus from his infected eyes. It was just awful trying to treat his eyes all day long. I remember at a point of desperation, about a year and a half old, writing this on the medical blog we kept for him as things got worse and worse. It became clear after some lab tests that he may only live to about 3 years old. I remember writing that there was this race between his heart, his brain, and his liver to kill him first. And so the physician said, you have maybe 3 years total here. And we'd already lost a year and a half of them. And we had a year and a half left, maybe. And I remember saying at the time, it looks like the window for miracles is about to close. We're going to hold on to hope. Now, of course, we'll do everything possible for him. And if that fails, we'll try the impossible. And fortunately, the impossible came in the form of the birth of precision medicine. And so this is what Bertrand's life has led to. And, I'm skipping over a lot now, but basically he was one of the first people ever to have his exome sequenced as part of a clinical trial. And with that, we got a diagnosis after 4 years. It turns out he was the very first patient in the world ever discovered with this brand new disease called NGLI1 deficiency. So that was a shock, but also a relief to just know what he finally had. And it also allowed me to make a rational prediction. Knowing that this gene is missing, throwing in a little more science, we could say, hey, it looks like he's missing something else. He's missing N-acetylglucosamine in particular. This is likely in deficit in him. Now, at the time, that word N-acetylglucosamine didn't mean anything to me, but I could Google it, and I did. And sure enough, there it was on Amazon. You could just buy it and have it shipped directly to your door. So I did. And as Sarah mentioned at the start, I actually took the entire bag myself, and effectively made this FDA phase 1 safety testing. Because I woke up the next day, and as you can see by my talking to you now, I'm not dead. I survived the trial, and that for me was good enough to consider giving it to Bertrand. So after wrestling with this idea of an N01 trial for Bertrand, I actually did give it to him. And I didn't know what it would do. My hope was that it would actually help with the epilepsy. It's not what it did in practice. It actually ended up helping with his vision because about 3 days onto this, when I walked into his room one morning, he was actually crying tears for the very first time. And that was really the end of his eye problems. We never again had problems with his eyes after this. It was just remarkable. And those little tears, they swelled ultimately into an ocean of science for the disorder as a whole. And I used to talk to biologists about this. And they would go, well, it's great that you're helping the people, Matt, but we're biologists, and what really matters to us is, can you cure the flies? I said, well, let me see what we can do. So I partnered with some fly biologists, and we made NGLI1 deficient flies, and sure enough, they die, pretty rapidly. About 18% survive. But if you give them this exact same compound, their survival shoots all the way up to around 80, 90%. So it's a pretty dramatic rescue for the flies. I don't know if the flies also cry, but they're alive at least, which I think is a significant improvement. So if you want to know more about this, it's written up, it's published. And then I went on, I wanted to know, can I do more than make Bertrand cry? I picked up a grant to do real drug development for this disease, partnered with folks at the University of Utah, where I was at the time, and got going. We did lots of computational screening for this, called docking simulations, really a lot of molecular interaction studies. We found 70 compounds out of 200,000 that we screened on a computer that looked like they might hit the right targets that we were after. And the good thing was 14 were already FDA approved. So we took all 14 to the bench, we tested them one by one, and one actually worked in the lab. And that happened to be an acid reflux drug called Prevacid. And so that was exciting. And we wrote the journey of that up too. It's also published now if you want to read more about the process that led us to that. But now you have to ask, well, what did all this do for Bertrand? Well, the good news is, it led to a much happier life. It in the end, led to a complete elimination of his seizures. So no seizures, no problems with his eyes, and lots of smiles every day. And I think the key thing for me is that he was never normal or perfect by any standards that a lot of parents would use, but he was normal and perfect to me, and that's all that really mattered. And he was happy. By the metric of smiles per hour, Bertrand was winning at life all day, every day. And in fact, he got to the point after a couple of years on this where his development had progressed where he could actually start using an eye gaze computer and expressing himself, which was astonishing. So about a year and a half ago, I walked in the door and he was able to blurt out, hello, like Matt is my dad, that was just Bertrand's way of saying, hey, welcome home, Dad. And but it was precarious, always precarious, because in May of 2019, I'll tell one story about some of Bertrand's life, but this is probably the darkest moment that we had, in the sense that it taught me just how fragile he was and how precious his life had become. So it started off taking him to the Star Wars exhibit at the local science museum in the morning. Checking out the aquarium, and 5 hours later, he's almost dead of septic shock. Almost no warning, almost no sense that this was coming. And I'm sure this is an experience that a lot of the parents watching have had, from normal day to in the hospital in total chaos in just hours, and launched a second diagnostic effectively on the fly. It's going, oh my God, what do we do? How do we solve this? Because it was culture negative sepsis, couldn't figure out what bug was driving this or how to treat it. And this turned into 5 weeks in the ICU, 5 weeks. There were pleural effusions, there was ascites, and just random precipitous declines in his health all the way through. And 3 weeks in, he looked like this in the morning, and by evening, he was almost gone. In fact, that night, we basically said goodbye and said, we think this is it. Didn't look like he was going to make it. Somehow he did. In the morning, his hematocrit was at 19%. He needed to get a chest tube to drain the pleural effusions. But to even get to that point, he had to get a blood transfusion now. They got it. They put in a chest tube, put it in the wrong place. So he coded. They took him back, put another chest tube, and this one actually worked, at least to get the fluid off his lungs. But the cultures were still negative. We still didn't know what this thing was. So going into week 4, Memorial Day weekend, I remember the resident coming and saying, well, nothing happens on weekends. We're just going to wait 3 days now. And I said, well, then I'm going to write some code. I got to do something. I've got to somehow figure out a way to help Bertrand. I ended up making a diagnostic extension to our artificial intelligence tool to go after infectious diseases as I sat by his bedside. And what it did here was it allowed you to put in the symptoms and try to figure out, the diseases that were most explanatory for all the symptoms that you saw. And it ranked them and said, okay, here are the top diseases that explain all the symptoms you see. And so it came up with this list. These are the top diseases that it thought Bertrand might have at the time. And we began very aggressive testing for these. And in parallel with that, also kicked off an effort in what we call metagenomics. So your metagenome is your genome plus the genomes of all the organisms floating around inside of you. And if you take any sample from a human, you're going to get mostly your DNA floating around in there, but you're also going to get DNA from the bugs floating around in there too. And this is a clue as to what could be driving an infection. So the question was, can we use that free-floating DNA from these organisms to figure out if one of them was the cause of the infection? I actually got his pleural fluid, his blood checked out at the pathology department at the Children's Hospital, drove it halfway to Huntsville, Alabama, where I've handed it over to my deputy at the Institute. He drove it the other half of the way to Hudson Alpha, which is Genomics Institute in Northern Alabama, and started 4 rounds of shotgun sequencing for his genome in these fluids, and his metagenome over the course of 3 days, something which would have been unthinkable 10 years prior. So in the end, this allowed us to figure out what it was. By looking at this, I was able to generate this picture, within 3 days of getting his blood up there. And it told us what was possibly, what was in his blood, and really more importantly, what was not in his blood. If you started crossing things off this list that just weren't there, there was actually only one thing left, and that was Pseudomonas. So it looked like an aggressive, unusual Pseudomonas infection. The solution in this case was then Meropenem, which he got, and after 4 weeks in the ICU, he was out within 48 hours, and his pleural effusions were receding, his ascites was diminishing, his O2 needs were dropping, and when another round of metagenomics on top of this showed that Pseudomonas was gone. So he was, just almost overnight from death's door to feeling better. And after 5 weeks, he was home and reunited with his pet fish, which he just loved dearly. And, just always a happy moment when he saw these fish. So, in fact, a week after that, I took him down to the beach for Father's Day weekend, and I got him swimming with the dolphins, because that's something I promised him we would do once we got him out. And I think he watched Dolphin Story like 80 times in the hospital. So I was like, we got to go swimming with the dolphins. But I realized from this that Bertrand, honestly, with a child with a life expectancy of 3, he was 11 by then. He was deep into bonus time. That's what I realized from that whole episode 2 years ago. I had to move faster. So that same month, I ended up getting a clinical trial funded by NIH for another experimental therapy for him in partnership with Mayo. I got him up to Mayo a few months later to actually start the trial and do the safety checks. And then the pandemic hit, slowed everything down. And he had a rough time. He ended up in the hospital multiple times, during the pandemic. And his health just started declining. Not from COVID, just in general. It was a rather precipitous decline, really, as puberty took hold. And I didn't know how much time he had left. I didn't think that there was necessarily an immediate end coming, but I knew that every day was becoming very precious with him. And so for several months, I took him on what you might call a sunrise tour. This was in the fall, and late summer, early fall. And so I just started taking him to beautiful places and looking at sunrises. So I took him down to the beach in Florida. We saw this together. To mountain lakes, saw this together. And then this here is, in fact, the last sunrise we ever saw together. This is from my childhood lake house home, where I went every summer as a child, and I took him up there. This is in the mountains of Georgia. Because a few days after that, and some pillow fights and some smiles, he was gone. There was really no warning. He went to the hospital. It was sepsis, and 48 hours later, after the briefest hospitalization of his entire life, that was it. It was just I just can't believe how fast it all happened. I was in the process as he was going in, saying, okay, let's do the metagenomics again. Let's figure out what this is. And before the results could even come back, that was over. Yeah, and I remember, looking for sunrises the next few days after that. And I was, the very next day I was driving by the park I used to walk him through all the time, and the sun didn't come up. There was a fog bank over Birmingham for the entire week, and there was a strange golden glow over everything. Yeah, and the city just looked like this for days. So there was no sun rising, which fit me perfectly because my sun wasn't rising anymore either. And I spent time thinking about, what do I do? What does this mean to me now, going forward? And I realized I want to double down on the mission. And I thought a lot about, the parents that are out there listening to this and what it means for you. And I remember I found something I wrote years ago about what it meant to be a rare parent. And I hope this resonates with you now too. And to me, what it means to be a rare parent is, it means fighting when you know you cannot win. It means believing the crippled will stand and walk. It means believing the mute will speak, and it means believing the blind will see. It's about never yielding to the impossible when your child is on the line. It is about exerting a force of will so strong that, if even only for the briefest of moments, the line between the possible and the impossible bends. It is about exerting a force that for so long and with such intensity that in rare circumstances, that line breaks. And so, for the parents out there fighting for your kids, I know this, this is you. And I applaud all of you for everything that you're doing for your children. And, it's given me a chance to think about what really matters. And I'm grateful that I have a job where I can spend all day, every day, actively living out the legacy and the mission that Bertrand gave me in life. And I realized that, I'm grateful that I had those happy moments with him, because at the end of the day, it really was the happiness that mattered. And he lived, if a brief life, at least a very happy one. That's the life I want to be able to create for all the patients that reach out to the Institute now. So I've created a specialized endowment at UAB where it'll be around forever. And as it grows, the interest each year will be used to fund science in the service of patients just like Bertrand. In fact, we've just successfully treated our very first patient with the interest from this fund. In this case, it was a rare genetic skin condition. We created a custom compounded topical cream with an EGFR inhibitor in it, and it actually worked to clear up this horrific case of acanthosis. So it's just amazing to me to see, a patient already benefiting from this. And if you go to his website, you will find a full 90-page write-up of this algorithm for precision medicine, really targeted at patients and parents that find themselves in the same position I did 13 years ago. So with that, I'm just grateful for the chance to speak to you all. If there's any questions, certainly happy to answer them. But just again, thank you so much for having me. Oh, I think you're on mute.
There we go. I'm unmuted now. Hi, thank you. That's probably a good thing because I was a bit teary, so I've got it together now. So see, I did that on purpose. Bertrand was really lucky to have you as a dad, and we're all lucky to have you in our corner. So thank you very much. When you talk about rare parents, I love Harry Potter. I love all the smart books, and I love Harry Potter, and I love how Harry learns from Luna Lovegood that he can see the thestrals that he never saw before. And when you have someone you lost, or when you have someone who has a very rare disease, I think you see things that other people can't see. And now you are helping us find treatments for them, and for that we thank you. So the first question is, Metagenome sounds way cool. So how do we access it? Is it available to ordinary people wherever they are around the globe?
Yeah, that's a great question. So it's what we call open source software. So it's freely available for anybody who wants to download it and run it. It's not user friendly in that sense just yet. You have to have a little technical sophistication to get it up and running. We're working on ways to get around that by creating a web interface that'll make things simpler. There are some licensing issues with the data sets involved. So if you're an academic, you can very easily get the data sets that you need for free. If you're a parent, honestly, the easiest way to get to use it is to email me. I can route you to our analysts, these students, and they can help run the queries for you. I think that's going to be the easiest way to do it.
That sounds like the smartest way to do it. But the question that I have is there a fee for that?
No,
Obviously you've done a lot. No, this is just.
no fee for that either. So the reason we try to do everything computationally is that it's just the cost of electricity at that point, which is nothing. So if we need to do something like build a model organism, we have some funds to cover some of that. But then we have to actually start charging for lab costs and things like that. But we're trying to make this as accessible as possible. And so free is an important feature of the service to the extent we can do that.
That is just monumental in and of itself. And it really strikes me, your journey is a reminder of how far we've blazed the path forward in the past years. For the people who are just starting their journey, I know there are people on this call, some who've got children who are my daughter Jackie's age. I mean, she's almost 17, but we've got some people in our community who are on this call now who might have had a diagnosis a couple of weeks ago or a couple of months ago, and they're struggling with the load of that, the weight of that. What would you say to them about what you've learned and where we are now and where we could be headed in the next 10 years, Matt?
Yeah, well, first of all, whether you've been diagnosed a long time ago or recently, my heart goes out to you, parent to parent, it's tough. I remember those early days when you just didn't know what was happening. And and the shock. What I would say is, have hope. Things are changing incredibly rapidly. I think about how fast things changed in the course of Bertrand's life, and what his quality of life was at the start versus what it was at the end, and it was a dramatic change. Bertrand was very happy all the way to the very end, with all the improvements that he was able to access that would have been unthinkable, at the time that he was born. And so I think it's a hopeful time, and I would have optimism and hope for any new parent or any old parent out there. Honestly, things are changing so fast.
I'm going to do a tiny bit of a jump cut. We've all around the world been through COVID, and we saw what happened with these vaccines, some brand new strategies that came around coming up with these novel vaccines, particularly mRNA vaccines. And I wanted to ask you, are there any lessons from that with your background in computer tech? Are there any lessons from that giant worldwide experiment when it comes to us in the path forward with genetic epilepsies and precision therapeutics?
There's a lot of lessons to learn from the COVID experience, even before you get to the vaccines. I mean, I think it shows that, if society feels something is important enough, then it turns out a lot of what we thought were obstacles are not obstacles anymore. And we can do things with remarkable speed. It also taught me the importance of open science. COVID was the first grand worldwide experiment in open science where suddenly the papers weren't locked behind paywalls and anybody could get to them anywhere, anytime, in real time. Everything was put on preprint servers. We didn't wait for them to get formally published. The acceleration in the science as a result of that was extraordinary. And that should be the norm for every disease going forward. I mean, absolutely. And then if you think about it, mRNA vaccines have suddenly made the era of oligonucleotide therapeutics, here. It's here. It was already happening. If you look at antisense oligonucleotides that were getting approved or used on an individual basis, it was beginning. But now we're seeing people inject nucleic acids in the cell left and right. Now, mRNA might not be exactly the right substrate for doing this for rare disease, although for some, it might actually be just fine. It might be all that we need. In fact, that was the original purpose of Moderna. They were going to try to do treatments for rare disorders, and ended up pivoting when it looks like you could do mRNA vaccines instead. But the idea of programmable therapeutics, I mean, this is big. This is very big. It has a lot of potential, and I think people are already looking at very novel ways to manipulate mRNA. I think the most interesting one is a recent one, where instead of delivering the gene editing proteins themselves, they delivered the mRNA for the gene editing proteins, and so it reconstructed the constructs inside the cells without having to deliver these challenging proteins. I'm like, so there's going to be a lot of interesting and creative developments that follow on from the actual clinical introduction of mRNA therapeutics.
Yeah, I think that's very exciting. And I love as well your point around the openness, the fact that you could get access to these papers and everybody was working together on something that was so big and which affected us all. I want to come back now. We have one more question regarding Metagenome, and it relates to hit rate. And I'm sure there is a little bit of a difference depending on whatever the particular question is, but can you give us a kind of a range of the hit rate that you have there at Metagenome?
Sure. Yeah, so it does depend on the kind of gene that you're going after. So I can tell you from experience, the highest hit rates are for cancer genes, just because they're so well studied. And so we tend to find lots of molecules that will, not because there are lots of molecules, it's just because they're most observed. Most experiments have been run on them. But as it pertains to ion channels, the sort of a very relevant tranche of genes, as I mentioned earlier, or epilepsies, ion channels are up there, and we do get hits for those quite frequently. They do seem to be amenable to pharmacological manipulation, and so I would say prioritize reaching out if you do have an ion channel driven epilepsy. Something might pop up. We can work with your treating physician if there's enough research to make it seem like a reasonable thing to do to get a recommendation out.
Okay, and I just have a couple more questions around that.
Yeah.
Yeah, in terms of that, I mean, specifically, just because, I'm imagining all the people on this call saying, what exactly do I do? Are you going to be looking for a specific medicine that they might be able to give their child, or a specific treatment protocol? What kinds of things would they be asking for if they're on this call?
So the full process is, once we've got the target, let's suppose we know we want to inhibit a specific target. We'll ask Metagenome, go find inhibitors. Some of the stuff that comes back, some of it may be drugs that are already approved, some might be things in clinical trials, some of them might be natural products, and some are things you would never even ever want to consider putting in a human being. And you'll get a different mix each time, depending on what you're going after. And then we prioritize based on our sort of estimates of safety and appropriateness in terms of the concentrations needed and whether it'll cross the blood-brain barrier. We've got a sort of a ranking process that we go through, and hopefully we can distill it down to one really compelling clinical recommendation. We will take that to an internal physician and say, if you got this research report, would you make a recommendation based on it? And if they say yes, we will send that report over to the treating physician, and then they can make the call on what to do.
What a phenomenal program. Well, it's very exciting research, and there's a small d democratic feel to the whole thing in that it's available so broadly. So thank you for that kind of very friendly version of precision medicine that uses AI to make something that's so much more broadly available to us all. And thanks as well for joining us from the US. You're in Alabama, Central Time. So what is it, about 9 o'clock now?
It's about 9 o'clock, yep.
All right, well, we're going to let you go and enjoy your evening. And thanks for the inspiration and the education on this important topic. We hope to see you again soon, Matt.
All right, thanks, y'all.